So remember that there is a short introductory video to animal body form and function. And so hopefully you had the opportunity to watch that. If not, you want to make sure that you watch it before you take the quiz or come in to take the quiz on Wednesday. So we're going to talk about histology today, and this is the study of tissues. So you can take whole classes in histology, and you can also study the difference between normal tissues and pathological tissues. So you can take pathophys, looking at the histology of damaged tissues or tissues that have become diseased. So embryonically, remember that we actually have, or we are actually, my hand is affecting this. We are actually what is said to be triploblastic. So what does that mean? Did anybody watch the video and know what that means? Yes.
3 layers.
Or embryonic tissues, close. So this is that we have 3 layers of embryonic tissues. So all vertebrates, for example, and actually most of the animals are triploblastic. We do see that some animals, like the sponges, lack tissues entirely. When we look at the jellyfish and the sea anemones, which we might get to today, they actually only have 2 embryonic tissue layers. And so this triploblastic means that all the other animals, including like earthworms, including clams and octopi, for example, and us, have 3 embryonic tissues. So these are unspecialized. So we can talk about them containing stem cells that are then going to become specialized. And this process is called differentiation. So interestingly, it's important to remember that we all start out as a single cell. And then that cell undergoes reproduction, undergoes mitosis, and we become a multicellular embryo. And then those embryonic tissue layers arise, and then they give rise to very specialized types of tissues. And so what that means is that all of our cells have the exact same genetic material. So this has to do with gene regulation. So if you have the same genome, for example, in a liver cell as you do in a skin cell, it has to do with how genes are regulated, how they are turned on and turned off determines that specialization. And we still have stem cells in our body. Where's a good source in our body of stem cells? You're going to find a good source of stem cells in your body. Where are they located? The bone marrow, right? Because our red blood cells, for example, lack a nucleus. And so our red blood cells cannot reproduce. And so we have stem cells in our bone marrow that undergo reproduction to continually produce red blood cells. Those stem cells could be taken out and used elsewhere. It used to be thought that there were only a few places in our body that we had stem cells, but now we're actually, every time we look at a new part of the body, like our fat tissue, for example, they just recently discovered that there's stem cells in our adipose tissue, in our fat tissue. And so it's kind of interesting to think about those stem cells. It's also kind of interesting that it seems that these stem cells can cross the placenta. So for example, you might have, if you have given birth to a child, you might have stem cells that from the baby that go into your body and then take up residence. So we'll talk more about stem cells when we talk about reproduction and development. But there it's important to realize that all of our cells have that same genetic material. So one of the important things about histology is that we have form. We say that form follows function. So what this means is that the function of the tissue is super important in determining what that tissue looks like. So for example, we can talk about tissue that is stratified. So we can talk about, for example, epithelial tissue. Epithelial, if I look at this word, epi means upon, and thelial means a layer. So epithelial tissue layers, so it's found on the outside of the surface, outer surface of an organism. It also lines body cavities. So all the way from the mouth to the anus in our digestive tract is actually lined by epithelial tissue. So a good example of how form follows function is we have stratified epithelial tissue. And this means that it has many layers. And so wherever we see stratified epithelial tissue, this means that it is protective. So its function is in protection, because what that means is that those layers can be constantly slept off and replaced. So it could be the outer surface of our skin. It could be the lining of our mouth, which is stratified. Our esophagus is also stratified. When we get down to our stomach and our small intestine, that is not stratified. And so we can look at, say, for example, simple epithelial tissue. And that means that it is not stratified. Let me see if I can write neater. Simple epithelial tissue. So this is a single layer. It's a single layer. So it is not protective. So it can have another function. So for example, it could function in the absorption of nutrients. So we have a simple epithelial tissue lining our small intestine. And so in the small intestine, this is absorption of nutrients. In our lungs, we have simple epithelial tissue where we need gas exchange to occur. So where oxygen and carbon dioxide is exchanged with the air in our blood, that would also be simple epithelial tissue. So this would be in where we have gas exchange. So these would be good examples of how form follows function. The function of being protective or exchange of substances, absorbing or gas exchange, would be simple epithelial tissue. So I'm going to give you a little handout here, and I would like you to see if you can match up the tissue with its function. So this is just a little matching. We're going to do the homeostasis, a little part of that, not right now. So you can see if you can do this. You could work with a partner, or you could work alone if you want to. So this is a little preview of looking at the different types of tissues and where they are located and what their function might be. So sometimes I included a function that might be a little bit different than what you might have seen before. So I'm going to give you a little handout that's got some different tissues. On the front side of it, it includes the different types of epithelial tissue. And then on the back side, it has the different types of connective tissue. Okay, so let's go over what you have so far. So what type of tissue is found in blood vessels and is responsible for vasoconstriction and dilation of those blood vessels? What do you think? I want to give a guess. No, most of you have these filled out. What was it? Nobody wants to guess. It is smooth. Cardiac muscle is the heart muscle, so it is smooth muscle. They can store energy and also function to cushion some organs. Adipose tissue, excellent. Most abundant type of tissue in the spinal cord. Nervous tissue. So the spinal cord actually contains some other structure, so it's actually an organ, right? But it primarily has nervous tissue. Found lining the trachea and helps to move mucus up from the lungs toward the throat. You could look at that handout that I just gave. Where is it in the trachea? Oh, no, it doesn't have that one, does it? Oh, it does. So in the trachea, this is the pseudostratified ciliated. So cilia is what helps to move the mucus. Where our bodies store calcium, where is calcium needed in the body? Bone. So that would be skeletal. Bone. So that would be bone, sorry. Contraction of this tissue, as in shivering, can help increase body temperature. What do you think? Skeletal muscle, excellent. Makes up the support rings in the trachea. Support. What do you think? It's hyaline cartilage, yes. Hyaline cartilage is actually the most abundant cartilage we have. Found in your external ear, this is also cartilage, but it would be elastic because it absorbs energy and then rebounds, right? Keeps its shape. Found in the outer layer of the skin or epidermis. Stratified. So stratified squamous epithelial, which would be right here. And then allows the ventricles of the heart to pump blood. That's cardiac muscle. Okay. So you'll notice here I've given you some different examples of epithelial tissue. One of the other characteristics of epithelial tissue is that it is characterized by cell shape and then the number of layers, which we already talked about. So if we're talking about epithelial tissue, we can talk about the way that it is categorized. And we can talk about layers, right? So we talked about simple and stratified. What about this pseudo-stratified? What does pseudo generally mean? False. And so one of the things you'll notice about this is it really is only one cell layer thick, but the nuclei are at different levels. So if I'm looking at it underneath a microscope, it kind of looks like it's striated because the nucleus are at different levels. We can also talk about cell shape. So squamous, you can just remember, is like being squashed, right? So it's a flat cell. So this means flat. And so, for example, that would be important for gas exchange because we simply want oxygen and carbon dioxide to diffuse across that surface. So that would be like in the lungs. The respiratory membrane is simple squamous. Okay. We can also see that a lot of them are columnar. So they're column shaped. And that is important when we're talking about digestion and absorption. So this would be like in my small intestine. Okay. So it is columnar, column shaped. And then I think there's an example of cuboidal. So cuboidal is actually found in the kidneys. So this is in the kidney tubules. So it helps with filtration. This would be the respiratory membrane. This one would be in the esophagus. So when you think about your esophagus, it has a protective function. It's not for absorption, it's protection. As food moves down through your esophagus, it would scrape maybe the esophagus a little bit. And so that is in protection. Okay. So you should be able to give me an example, specifically with the epithelial tissues, of how form follows function. What does that mean in terms of the structure of the tissue? Does anybody have any questions about that idea? Form following function. Okay. The next type of tissue that we're going to talk about, we're still talking about the epithelial tissue because epithelial tissue gives rise to glands. So this gives rise to glands. And glands are substances or are cells that secrete substances, right? So when we talk about glands, we can talk about the two different types. We can have endocrine versus exocrine. So which one of these glands produces hormones? An endocrine gland or an exocrine gland? Endocrine. So endocrine secretes hormones into the circulatory system. So hormones are just released into circulation. This is just not working right. Let's see if I can. It's my magnetism today. Hormones secreted into the circulatory system. So these types of glands are said to be ductless. As opposed to exocrine glands, these secrete substances into a duct. What is an example of an exocrine gland? That duct then transports the substances to where they need to be. The adrenal gland is actually part nervous tissue and it's part endocrine. A good example would be like salivary glands. We can also talk about mammillary glands, which produce milk. So these contain cells inside that secrete their substance, but it is transported via a duct to the mouth or to the nipple, right? Or in some cases, we'll talk about platypuses, which actually just secrete milk onto the surface of their belly. So they actually don't have nipples, but it's transported via ducts. Sweat glands would also be another example of exocrine. Now there is a organ in your body that has both. So the pancreas has both endocrine and exocrine function. So what is an example of a hormone that the pancreas secretes? Insulin. So that would be its endocrine function. It also produces an antagonistic hormone called glucagon, which helps to coordinate glucose levels. It also produces pancreatic enzymes. So these enzymes are produced by the pancreas, and we'll talk about how they're transported to the small intestine, and they actually help to break down lipids and proteins and carbohydrates so that they can be absorbed by the lining of the intestine. So the pancreas is interesting. It's epithelial in its origin, and it is both an example of an endocrine and an exocrine gland. Our master control gland is actually the pituitary gland. So we'll talk about how the pituitary is coordinated with the brain and how it produces hormones that are secreted and help to regulate our cycles, including our metabolism, like the thyroid gland. Okay, so we actually have an example in this diagram of what is referred to as a unicellular gland. And this is called a goblet cell. It's called a goblet cell because of the way it looks. So it's got a narrow bottom, and then it forms into this larger top, like a goblet. Okay. So when we look at the goblet cell, it produces mucin. And this mixes with water to produce mucus. So when we think about mucus, we think about being sick, but really we're constantly producing mucus, and it's really important that we do. Mucus is really important in the digestive system. When we talk about like the lubrication, so its function, mucus functions in like lubrication. In the stomach, it's super important because it protects the stomach from acid. We also obviously have mucus that we produce in our respiratory system, and that captures microbes and debris. So like by breathing in through our nose, we're actually filtering the air of debris, and we have mucus up there, and cilia that helps to capture that debris, right? So this doesn't actually go into our lungs and cause damage to the inside of our lungs. So mucus has important functions in the body. And so it is in the epithelial tissue as a single cell. So this would be like, for example, in the lining of the trachea, because we see the cilia, and then we see the mucus on the surface. I'm going to skip that one. Okay, so the next type of tissue that I want to talk about is the connective tissue. And the connective tissue is very diverse. And all the connective tissues are derived from mesoderm. So they all come from the same embryonic tissue layer. So that would include like your bones, your cartilage, your fat, your blood. Those are all examples of connective tissue. So this is very diverse group. So if we look at some of the distinguishing characteristics of connective tissues, it would be that we have cells within a matrix. And the matrix is not composed of cells, but it's produced by the cells that are within the matrix. So when we talk about the cells and the matrix, let's actually talk about the matrix first. So the matrix is acellular, and it consists of a ground substance. So the ground substance is mainly composed of water. So it has water in it, and it also has what are called proteoglycans. And so if you break that word down, proteo means proteins, and these are proteins that have sugar molecules attached to them. And this ground substance actually, in the case of the matrix, actually traps water. So this functions to trap water. So I'm not going to talk about blood right away. I'm going to talk about other types of connective tissue. The blood actually has a plasma. The matrix is actually the blood plasma. So we're just going to talk about the other types of connective tissue, which is sometimes referred to as, how do I want to say this? I don't want to talk too deeply about it. We'll just talk about the main type of connective tissue. So we also have fibers. So if we're talking about like the connective tissue underneath our skin, or if we're talking about our tendons, our ligaments, our bones, we have fibers. And so the fibers are proteins. And there's two different types of proteins. The first one is called collagen. So you might have heard of collagen injections underneath your skin, because your deeper layer of your skin, your dermis, actually is made of connective tissue, and it has lots of collagen fibers in it. And as we age, we lose collagen, and so our skin begins to wrinkle, and it becomes, your skin kind of starts to be less firm because of the lack of collagen. Okay. So collagen is an example of a structural protein, and it is white in color. So this is why, for example, our bones are white. This is why tendons and ligaments are white, is because they have those collagen fibers located in them. We also sometimes have elastic fibers. These are darker in coloration, right? And they give the tissue its flexibility. So when we're talking about the blood vessels of the major body, like the aortas, the aorta has some elastic fibers in it that allows it to expand when the heart contracts and then to relax when the heart relaxes. So we can see elastic fibers. We also have elastic fibers in the connective tissue of our ears, for example, and also in the tissues that are the connective tissue that is our epiglottis has elastic fibers in it. Okay. Then we have the cells. And the cells are generally named according to what they are found in. So, for example, we can talk about osteocytes. Where do you think osteocytes are found? In the bone. So osteo, like osteoarthritis, this means bone. We can talk about chondrocytes. Where do you think those are found? What makes up the rings of our trachea? Cartilage. And then we have like fibrocytes. And this would be like loose connective tissue. That might be found underneath the skin. They're called fibrocytes. And then we might also have cells that are called erythrocytes. What is an erythrocyte? Does anybody know? Erythro means red. And so these are actually red blood cells. Oops, red blood cells produced by bone marrow, which is also connective tissue. And so what is the technical name for white blood cells? Leukocytes would be the white blood cells. We also have adipocytes. Where do you think adipocytes are? What is adipose tissue? What type of tissue is that? Fat. So this would be fat cells. Adipocytes. Okay. So on your handout are some examples of those types of tissues. So we have, for example, loose connective tissue. The loose connective tissue is actually very, very common in the body. So like we have loose connective tissue underneath our epidermis. It's everywhere. We also have loose connective tissue that is found between tissues. And sometimes that loose connective tissue as a name is called fascia. So when you talk about myofascia, that is the connective tissue that is around the muscle. Here you can see the cartilage, and that should say chondrocytes. Is that right? Did that one not come out? Okay, so for some reason this image got screwed up. So that should be CHONDR, chondro, and there should be a Y in there. And then we have chondroitin. There's an H there. Chondroitin sulfate, which is part of our ground substance in our cartilage. Where have you seen chondroitin sulfate before? You can go to Bi-Mart or Safeway or Rite Aid and you can buy that. And why would you buy that? Does anybody know? Those are said to be joint nutraceuticals, right? So you could buy it in hopes that if you ingest it, it'll make its way to your joints and will actually make your joints more cushy, because that's an example of a molecule that traps water. And so some other things that you might hear of when you have joint problems is you might take glucosamine, right? That's another precursor to a proteoglycan, like hyaluronic acid you might take, right? They even are prescribed these to dogs that have joint problems, right? You give your dog a joint nutraceutical and hope that that will somehow make the connective tissue more healthy. Okay. Here we have fibrous connective tissue that's got a lot of collagen fibers. So that would be an example of what? What does it look like? What type of structure is that? Does anybody know? It's a connective tissue connecting muscle to bone. This would be an example of a tendon. Adipose tissue, this is fat. One of the really interesting things about adipose tissue is that when we're adults, we do not lose fat cells. Even if we lose a lot of weight, we still have the same number of fat cells, but the fat cells can shrink up, and then they can fill up with lipids and expand. The only way to get rid of fat cells is through liposuction, right, where they go in and they suck the fat tissue out of your body. These would be the red blood cells versus the white blood cell. This should say plasma. So I don't know why this got so screwed up. And then this is the bone. Okay? So bone is living tissue. It's constantly being broken down and replaced according to the forces that are exerted on it. So that's why if you go up in outer space and you're not subjecting your skeleton to the right force, your skeleton will actually become weaker, right? It'll become lighter because you don't need a heavy skeleton in that particular environment. Okay, so these are examples of connective tissue. So this is that loose connective tissue. This would be the fibers. And the fibroblasts here show the nucleus. Those are the cells that are embedded in the matrix and are actually producing those fibers. And this is the bigger ones are the collagen fibers, and the darker ones, smaller ones, are the elastic fibers. Okay. The next category of tissues would be the muscular tissue. And so we can describe muscular tissue based upon its appearance underneath the light microscope. So we can say that it is smooth versus striated. So when you look at the smooth muscle cells, you'll notice that they just have one nucleus, and the smooth muscle cells are generally in layers. And so the smooth muscle is involuntary. So it's not something that we can control. It would be layers of cells. So for example, in our digestive tract, we have a circular and longitudinal layer. Right? So when the circular contracts, it actually causes the lumen, the opening in our digestive tract, to become smaller. And then with the longitudinal layers, it causes a shortening of the digestive tract. So when we have those working in coordination with one another, the circular and longitudinal layers, we call this peristalsis. And it's super important in the digestive system. If your digestive system stops moving like a wave, then the doctors start to get really concerned. And what they'll make you do is they'll make you get out of bed and walk around after a surgery to try to get that movement of the digestive tract to work again. So peristalsis would be an example of the movement of smooth muscle in the digestive system. When we look at striated, we actually have two different kinds. We have cardiac muscle. This is the type of muscle that is found in the heart. It is a really interesting structure because if you think about it, the heart has to contract against itself. It's not contracting against a skeletal element like bone, like the skeletal muscles do. And so it is characterized by having these intercalated discs. These are little structural elements between the cardiac muscle cells so that the cardiac muscle cells can contract and pull other cardiac muscle cells together to cause the contraction of the heart. So we're going to talk about this. This is primarily involuntary, although there can be biofeedback, right? So if you want to slow down your heart rate, you know how to do it. You sit quietly and you breathe really deeply and you calm your mind, right? And you can slow your heart rate down. And then we have skeletal muscle. So I'm just going to put that one down here. So this is skeletal muscle, and it is very interesting because it consists of multinucleated cells. So these are long, multinucleated cells. So the way that they get their multinucleation is they have fusion of stem cells. So when this tissue is forming from mesoderm, cells fuse together to form long strands of striated muscle. And these are the only really voluntary muscle. Right? This is what allows for movement. But a lot of our skeletal muscle actually also works involuntarily through reflexes and through reflexes. So for example, when I move, when I walk, I'm causing a conscious contraction of the muscles that move my leg, but I'm not paying attention to the muscles that are keeping me upright, right? So as I shift my body weight, there has to be all these spinal reflexes, which are not voluntary, that keep me from falling down. So to keep my balance, there's all these involuntary actions. So it's a voluntary muscles, but we can have autonomic function, right? So it could just be automatic through reflexes, but they are said to be voluntary. So muscular tissue is able to contract because of proteins found in it. So the ability to contract is due to proteins. Does anybody know what two proteins are specifically involved in the shortening of muscle cells? What are they called? They have a name that you might have studied before in biology, high school, or elsewhere. They are called actin. Has anybody ever heard that word before? And myosin. So we're going to talk about how these proteins allow for muscle contraction. The reason why we eat the skeletal muscles of other animals is for its protein, right? That's the reason why we eat chicken legs, right? Steak is that meat is high in protein because of these two things. We could eat heart, right? You can eat glands, you can eat liver, right? But the protein is found in the skeletal muscle. Okay. And finally, the last type of tissue is the nervous tissue. And the nervous tissue consists of specialized cells called neurons. And these neurons have cytoplasmic extensions. And these extensions create connections with other neurons that are called synapses. So this allows for connections between neurons. So for example, when we learn something new, we have synaptogenesis. We have the formation of new neural connections, and that's called synaptogenesis. And so if we look at the neurons, they're characterized by having a long cytoplasmic extension that would create a connection with another neuron. This is really important because information flows down the length of the neuron, and then at the end of this axon, which is a cytoplasmic extension, is the release of chemicals. And these chemicals that act as communication are referred to as neurotransmitters. So they release neurotransmitters. Now, it used to be thought that nervous tissue just had neurons in it, and that was the most important part of of nervous tissue. But we also have discovered that we also have glial cells. So it also contains, nervous tissue also contains what are called glial cells. And these glial cells might actually have a really super important function, and they might be the key to things like Alzheimer's, or maybe even the key to diabetes, for example. These glial cells are helper cells, and they actually outnumber the neurons in some places by 10 to 1. So for every one neuron, there's like 10 glial cells in the tissue. And so they're showing this as an example would be the astrocyte. And it's called an astrocyte because of the way that it looks. This would be in the brain. The astrocyte is star-shaped. And it actually protects the neuron by regulating its external environment. So if chemicals get into the space of the brain, they get across that barrier, the blood-brain barrier, then this is the glial cells that are going to regulate that internal environment. And then we're going to talk about these because these cells, the oligodendrocytes in the brain, produce myelin. And myelin is like insulation. And you might have heard of Schwann cells, which are very similar to oligodendrocytes, but they're found in our peripheral nervous system. So nervous tissue, glial cells are associated with nervous tissue. Okay, so we're going to talk about homeostasis as kind of an overlying, really important part of being alive. And this is the maintenance of a relatively constant internal environment. Homeostasis technically means same state, but notice that I put relatively constant here because there's this new idea that instead of homeostasis, we might want to call it a dynamic equilibrium. So this is kind of the same thing as saying a relatively constant internal state, because most of the time we're at equilibrium, but we need to adjust if there are changes in our environment, right? We don't want to completely stay always the same amount of glucose in my blood. So if I always have the same amount of glucose in my blood, then when I'm exercising, I'm not going to have enough glucose to power my muscles, right? I need to release glucose, and that glucose needs to increase when I'm exercising, right? So we're going to look at a video, and then we'll take a break. But the video talks specifically about heart rate at rest. So an example of this is heart rate at rest. Okay. And this is only like a 5-minute video. So you want to really think about how this is an example of a dynamic equilibrium. So they're taking heart rate over long periods of time.
Brian Goodwin sent me to see Ari Goldberger, a cardiologist at Harvard, with some very surprising ideas about health.
It appears that health represents a remarkable balance between excessive order on the one hand, things being overstructured, and complete randomness on the other, where there's, in essence, there would be physiologic anarchy. And the healthy systems like to be there. They don't sit still. They're always kind of fidgeting. They're always ready for everything. When people listen to a healthy heart, what they hear is something that sounds quite regular. It's either the lub-dub, or they are looking at a cardiac monitor that's showing something that seems to be ticking off impulses much like a clock. But in fact, when you look at the way the heartbeat is actually changing in a very subtle way, what you see is this extraordinarily complex variability. It was, to us, an amazing surprise to see that the resting heartbeat was in fact as complicated as anything that we were able to find elsewhere in nature.
If you measure the beat over many hours, you discover that the heart's rhythm is constantly changing.
Well, I'd like to show you a little game. It's a physiologic game that we developed in the laboratory. And what we're going to see is the pattern that your heartbeat makes if you're just basically sitting around.
This is just a resting heart?
Yeah, is that a surprise to you?
It is.
Yeah, this tracing here is very different from what you might suppose if you
Completely
came from the tradition of homeostasis. This should be, if you're resting and you're quiet, this should be a straight line or something approaching
Exactly.
that. So one of the ways of trying to convey what healthy physiology is all about is to take this sequence and to translate it, to transpose it or to map it into a sequence of notes. And so the question is, what does your heart sound like? What is the music of the healthy heart?
So that was wonderful. That's a normal heart.
That's a normal heart. And what we're going to see here now is a very different pattern. This is the heartbeat tracing from the patient with heart failure, and what you see here is actually quite different from the healthy. Now, things actually look more organized. They look, in a sense, more periodic. And so let's play it out and see what this sounds like. So which would you prefer to listen to, the healthy heart or this?
Boy, what a contrast.
So very repetitious, very monotonous, and the sicker and sicker people get, the more the variability collapses, and ultimately you may end up with just one note repeating itself. So in the sickest pathologies, you can literally go from this very musical, tuneful dance, which is the healthy heartbeat, to what becomes a one-note marching band. The essence of healthy function is adaptability, the ability to cope with an environment that's going to play tricks on you. You don't know what's going to happen next. So having this sort of bubbly type of turbulent-like dynamic, which has all these different frequencies, all these different responses built in, gives you an advantage.
So as long as you're playing a symphony in your chest, everything's fine.
It's the old story about a song in your heart, but literally that is in fact the case.
Okay. So that's the idea of dynamic equilibrium. So we have to be able to adjust to the environment, right? So for example, with heart rate, some people, because they have high heart rate, so tachycardia, they have constantly high heart rate, sometimes doctors give them what are called beta blockers, which means that the nervous system is blocked from increasing the heart rate. And so a person who's on beta blockers, for example, might not be able to adjust their heart rate when climbing a set of stairs. So for example, they'll have to stop. They'll just my heart rate will not keep up with this exercise, this activity. And so that would be an example of not being able to maintain homeostasis when they had heart rate of 200, which could lead to cardiac arrest. And then when they're given drugs for it, if the drugs are not quite monitored properly, then that causes problems as well. And so this idea of this dynamic equilibrium means that it's a lot harder to understand or to regulate healthy physiology than you might predict. Okay, so we're going to take a little break here, and then I'm going to talk about the mechanisms of maintaining homeostasis. So we'll take maybe a 5-minute break. If you want to, and you understand homeostasis, you could look at that concept map that I gave out, which was on the back of your tissues. So it was right here. Oops, back of tissues. It'd be right there. was just texted a picture from my children, and my 13-year-old decided to paint the outside of the back shed because she was so bored. Paint. It's never going to dry. Paint. Why would you paint the outside of the shed on a day like today?
Is it a nice color though?
It's white. It was kind of a nice gray. And she used indoor paint. Because of course, we don't have.
Is she proud of herself?
Oh my God, it's her new fort. See, she's making a fort. Yes. So she took everything that was of importance to me out of the shed and then painted the outside of it. It's her new hangout. Yeah. Too bored. Okay. So, homeostasis. When we look at the mechanism, so homeostasis is the goal. So we need a mechanism to help with that goal. Right. So the mechanism has a name that's a little bit confusing. It's called the negative feedback mechanism. So when I write this down, people think that the negative, it must be bad, must be a bad thing. But it's actually a really good thing because it has the response in this mechanism actually decreases the stimulus to help maintain equilibrium. So the way that this works is that we have a stimulus, and it's important to realize that this stimulus could be internal as well as external. So it could be like you have too much CO2 in your blood, or it could be you are too hot, or you're touching a hot stove, for example, or you're listening to a sound. That could be the stimulus. So the stimulus is going to be received by a receptor. So the receptor is a modified neuron. So we actually have receptors on the inside of our body. So for example, your carotid artery has receptors that can detect CO2 concentrations in your blood. You have receptors in your medulla oblongata that can detect if there are chemicals that are in the blood that you want to get rid of, and that can actually cause you to vomit to get rid of those chemicals that you're absorbing in your digestive system. So this receptor needs to send a signal to the control center. So the control center often is the brain, but it's not always the brain. So when we're talking about the release of insulin, it's actually the pancreas that controls the release. It's not the brain that signals the release of insulin. So this pathway is called the afferent path. And I just remember that A comes first because from the control center, when it sends a signal out, this could be hormonal or this could be nervous. It's going to send it via the efferent path. And this is going to go to the effector. Okay. So the effector is going to cause the response, and this is going to decrease the stimulus and help to maintain homeostasis and equilibrium. So hence that decrease in the stimulus is why this is called a negative feedback. Negative feedback. This is actually the way that thermostats work, if they're working properly, right? You set your thermostat to 70. It senses that it has gotten to 68. It turns on. It produces heat, and then that heat, that response, then decreases the stimulus, and then the thermostat turns off, right? So it's much better than you just turning the heat on when you're cold and turning it off when you're hot, because it helps to regulate homeostasis. Okay. So this is different than a positive feedback mechanism. the positive feedback does not maintain homeostasis. So it is actually kind of a bad thing. Although there are 2 examples of positive feedback in our systems that are commonly used in your textbook at least. And this would include childbirth and contraction of the uterus. So for example, when the uterus contracts, it causes the baby's head to press on the cervix, and that pressing on the cervix actually causes more contractions and more forceful contractions, faster contractions. And so this builds until the baby is born, right? So there is an ending to it, but the contractions get more and more and more and more intense until the baby comes out, and then the contractions die back down again. So that is an example of positive feedback. And another one is blood clotting. Although again, once that blood stops being lost, hemorrhaging from the inside or the outside, we want the clotting to stop, right? So blood clotting also is a positive feedback. The more platelets that arrive, the more chemicals they produce, the more platelets are attractive, right? So that causes a positive feedback. Okay. So in your textbook, you have one on body temperature. So with your body temperature rises, it can send a signal out to the blood vessels. So in this case, the blood vessels would be the effector. And this causes blood to rush to the surface of the body. The sweat glands would also be an example of the effector. And then the response is that you have evaporation where heat is lost from the body. And then that causes a decrease in the stimulus. And then we talked about, or when we were looking at our tissues, we can conserve heat. We can actually produce heat by shivering. So shivering skeletal muscles would be the effector. They would cause chemical energy to be turned into mechanical energy, and that generates heat. It's an exothermic chemical reaction, which helps to warm the body, so heat is retained. So this is just an example of that negative feedback mechanism. And then this shows the positive feedback mechanism, which actually has to do with the production of oxytocin, which causes stronger contraction. So as the baby presses on here, it causes more oxytocin to be produced, which causes stronger contractions, which causes more oxytocin to be produced. And so labor would progress by increasing the intensity of the contractions. Okay, so let's look at the handout. I thought I put that on here, but show up. We'll talk about bioenergetics in a minute. So the handout that was on the back of your sheets. Oh, no, I'm not going to do this right away because we'll do this later. Sorry. I forgot, it didn't copy the things that you could put in there. If you love puzzles, this would be an awesome puzzle to do. Okay. But I actually had terms that I wanted you to put in there. So we'll do that later. Okay. So we can talk about, when we're talking about temperature and metabolism, we can talk about what is called bioenergetics. And so this is how an organism uses its energy. And sometimes this is called a budget, right? You have an energy budget. How much energy you take in, how much energy you expend. What do you use your energy budget on? So when we talk about organisms, we can talk about what are referred to as endothermic organisms or endotherms. So sometimes this is mistakenly or kind of misspoke in that you say that they are warm-blooded. However, ectotherms like snakes can also be warm-blooded, but it just depends upon their environment. So an endotherm really is an organism that uses metabolism to regulate its body temperature. So mammals, for example, are endotherms, and so are birds. There's some questions as to whether or not the dinosaurs that went extinct, if any of them were endothermic. And there's some question, crocodiles and alligators are almost on the verge of being endothermic. So some of the other reptiles can be endothermic. There's actually also, they just discovered a bony fish that also moves its fins in such a way that it generates heat. And they think that doing that is regulating its body temperature. So and also I can think of insects. When they quiver their wings, they're actually trying to warm up so that they can fly. So, but typically we say that endotherms are mammals and birds. We therefore have then ectotherms, and these use their environment to regulate their body temperature. So this is considered to be like fish, if we're talking just about vertebrates, amphibians, most reptiles. So they have behavioral thermoregulation. So oftentimes in the spring you might see lizards setting themselves on rocks or snakes going to the road to warm up, because they need to move into a warm environment in order to warm up, in order to be more active. So we sometimes say that these are just two alternative strategies, because really there is a cost and a benefit to each. So when you look at the cost and benefit of being an endotherm, so if we look at the cost-benefit analysis, we're talking about economics again, budgets and analysis. The cost of being an endotherm is that you need more energy. So that means you have to be better at capturing food, right? You have to be able to eat regularly. So, for example, people that have big snakes, like boa constrictors, they might need to only feed their snakes like once a month. Right? It takes a long time for them to digest anything, but they don't actually have a really high metabolic rate, so they don't need a lot of food, and it doesn't need to be regular, right? So the other thing, the benefit though, is that they can move around and they can inhabit and be active in colder temperatures, right? So can be active in colder temperatures. Right, so you don't see like large lizards in northern climates because it is just too cold for them to be active and to be able to get their food. Sometimes this is a problem. Sometimes if you live in a really cold environment, maybe you don't have food, maybe you need to hibernate as part of your strategy. So if you're a bear, for example, you would hibernate in order to be able to live through months where there is not a lot of food. So it's not the cold, it's mainly the food that they don't have in order to survive. So if we look at some interesting diagrams of this, you can see the dramatic difference in the energy budgets of different organisms. This isn't the one I want to use, though. Okay, so this would be endotherm. So this is a human. She's also, I think, pregnant. So they put reproduction as a proportion of her energy budget. So notice that just our basal metabolism, this is just staying alive, this is not doing anything else, is a pretty large budget. If you compare this to a snake, this is a much smaller proportion of the budget, right? Activity, growth, thermal regulation. So notice how much energy in terms of kilocalories per year that a human would take compared to a snake. So it's only 8,000, so there's much less of it as well. So they can expend a greater proportion in reproduction than, say, for example, a penguin. Notice, what do I want to say about that? What is this orange? That's thermoregulation. So notice something interesting about the mouse. Notice their thermoregulation budget. So notice that the mouse has a higher, it has to use more energy to regulate its metabolism than, say, for example, we do. So that takes me to the other diagram. Oops. Yes. So this is the amount of oxygen. They use this to estimate metabolism, so the amount of oxygen used. And you can see here that we have, as we increase in body mass, notice that that makes sense that an elephant needs more oxygen compared to a mouse or a shrew. That makes sense because of the size, right? But what if you took into consideration how much oxygen, how much metabolism per unit of body mass? So if we look at this other graph, you'll notice something really interesting comes about. So this is per kilogram. So this is the amount of oxygen per hour per kilogram. So notice that the shrew has a much higher metabolic rate if we take into consideration that how much oxygen it needs per unit of mass of its body than an elephant. So what can anybody think of why we see this difference in shrews compared to larger endothermic animals? Notice these are all endotherms. They do move around a lot, and they also have a higher metabolic rate. So like their heart, well, that's the metabolic rate, but their heart also beats faster, right? That's a good answer. Can anybody think of anything else? If we're talking about thermoregulation, surface area, right? So the smaller the volume, right? So small size of endotherms have a higher surface area to volume ratio. Okay, so it would be the surface area divided by the volume would be much greater in the shrew. So they're constantly losing heat. So if you're endothermic and you're small, you're going to need to have a huge metabolic rate. Okay? You're going to need to be able to eat. You're going to generate a lot of heat. You're going to lose a lot of heat. So as we increase in size, what we see is that this has a lower surface area to volume. So larger, lower surface area to volume ratio. And what this means is that they are going to lose less heat to the environment across the surface of their body. So shrews are notorious, like if they don't eat in a day, they're dead, right? They have to eat and eat and eat all the time in order to maintain that high metabolic rate. So this is another constraint when we're looking at endotherms. Specifically when we're looking at endothermic organisms, is that size plays a big role in their bioenergetics. Any questions about this idea? Okay, so we're going to start, and we're a little bit ahead, but we're going to start talking about the different groups of organisms. And so remember that I mentioned, and we watched a video about sponges and how sponges are the most primitive animal. So they are actually in the kingdom Animalia. Does anybody know what phylum they are in? Anybody learn this in? They are pore bearers. They have pores, and so they are porifera. So they are in the phylum Porifera. So that means that they are pore bearers. The other things that we talked about is that they lack tissues. But we do have cell specialization, which we'll talk about in a second. So they do not have nervous tissue or connective tissue or epithelial tissue. They are just aggregations of cells. They also have no symmetry. So even though they might look like they are radially symmetric, generally they are not said to be symmetric. So you could cut them in planes, but there would not be two identical halves. So they're said to have no symmetry. Some of them can get to be very big, like you could have those big barrel sponges that you could actually crawl inside. There's also freshwater versions of sponges, although most species are marine. So we do have freshwater sponges as well. So when we talk about the cell specialization, we can talk about the different types of cells that are found in the sponge. And I mentioned that we have cell specialization. We have collar cells, which are called choanocytes. These are involved in feeding. So these organisms are filter feeders, so they're feeding by filtering water in through these pores, and then the choanocytes are found on the inside. They're lining the inside of the sponge, and it is their flagella that create the current. If we look at some of the other cells, the porocyte, for example, creates a pore. So that is where water enters. So that's what the porocyte. So this opening is created by a single cell. The skeleton of the sponge is produced by a cell called the sclerocyte. So this produces what are referred to as spicules. So we'll look at some sponges in lab. Loofah sponges are not animals. They are actually the cellulose of a gourd. But if you're into art supplies, oftentimes you can go to the art store and they will have real sponges that you can buy. It used to be that they had a lot of, it used to be they were cheap. But now like one little tiny real sponge might be like $40, right? Because of their depletion. But they're oftentimes used in art. So the spicules can be composed of different things. They can be composed of protein, and that makes the sponges soft. They can also be composed of silica, and that makes the sponges rigid so that they would be like glass sponges. So there's different types of spicules and sclerocytes. We also have amoebocytes. And they're called amoebocytes because of the way that they move, but they distribute food. So they would function like the circulatory system in delivering nutrients to other cells. The outer part of the cell is called the pinacocytes. or the outer part of the sponge. Okay. So this covers the outer part of the sponge. So one of the really interesting things about the sponge cells is that it is believed that you could take a single amoebocyte from a sponge and it could develop into an entirely new sponge. And so they have lots of regenerative abilities. You saw the scientist was cutting pieces off the sponge and she said they would regenerate. So they have very high regeneration even though they have the cell specialization. So they have the ability to regenerate pieces of themselves. And this is actually the mechanism for a type of reproduction, which is called asexual reproduction. So we can talk about asexual reproduction. So this means without sex. So this would be like cloning. This would be sometimes like budding. Some animals bud and they produce a clone of themselves. And so this is where the offspring are genetically identical to the adults. And so sponges actually reproduce asexually when they're stressed. So specifically if they're stressed by water, say for example, the water starts to dry out where their tide pool is, might start to dry out, their pond might start to dry out. When they're stressed, they produce packages of amoebocytes. And these amoebocytes can withstand dehydration. So they can withstand drying out. And these are actually called gemmules because of the way that they look underneath the microscope. They look like little gems. They're kind of shiny and bright. So the gemmules would be the packets of amoebocytes. And so if we look at an image of this from a microscope, I'll come back to this slide. These are my gemmules. Right? So they're actually kind of embedded in, these are the spicules. This would be like a glass sponge. And these are packets of amoebocyte. And one amoebocyte can give rise to an entirely new sponge. So once, if this is a pond that's dried out, once it fills back up with water, those packages would break open and the amoebocytes would be produced, but they would be identical to their parent because they are diploid cells that are identical to the parental sponge. So they also reproduce sexually, so sexual reproduction, and this is the production of offspring that are different. So offspring that are not genetically identical. So I'll put genetically distinct. So interestingly, the amoebocytes can turn into either egg or sperm. The sperm are released into the water, and the eggs are retained within the sponge. So the sperm are released, the eggs are retained. So the sponges use the same mechanism that they do to obtain food as they do to obtain sperm. They filter it. And so if you imagine there's all these different organisms in the ocean that are releasing sperm into the water. So there's probably a lot of species of sperm out into the water. And even different species of sponges release their sperm into the water. So the sperm are obtained by the choanocytes. And the choanocyte can detect that a sperm is of the same species. So the choanocytes filter the sperm of the right species, and they pass the sperm to an amoebocyte. And the amoebocyte delivers it to the egg. So this is kind of amazing because it's such a simple animal. Why they would have internal fertilization? You know, even salmon have external fertilization. So this is internal fertilization because it's inside the sponge where the egg penetrates the egg, and then it develops into a mobile larvae. So the mobile larvae is released into the water. And this is very characteristic of an animal life cycle in that the sexually produced larvae are mobile. They can swim, right? And the reason why is that they can swim into an entirely new environment, settle down, and then become sessile, right? They can become sedentary once they've gotten into the right environment. So this means that they can swim out into new environments. So this is, they swim out into the new environment before becoming a sessile, which means not mobile, adult sponge. Okay. So if we look at a diagram of, or a little image of this right up here is my mobile larvae. Notice how simple it is. It's just multicellular. It's actually, this is shown two-dimensionally. It would be like a water balloon. So it actually has fluid inside versus outside. And notice it has cilia. So notice it can swim into the new environment and settle down and produce a whole new individual. Okay, so we'll stop there for today. And don't forget to do your, oh, actually, we have class tomorrow. We have lab tomorrow. I was thinking it was like end of the week. Don't forget to eventually do your Canvas homework for this week. And I will hopefully see you tomorrow.